Hey, what's up, and welcome to Blood Lecture number 2. Last time we went through some of the basics of blood, talked about plasma a little bit, talked about some of the things in there. What we did not get into is the formed elements. So we got to knock out the formed elements today, or at least two of them. At least 2/3 of the formed elements today. Number one, the one you see pictured in the drawing, the cartoon right there. It is a red blood cell, also known as an erythrocyte. And there's a couple awesome things about a red blood cell. Structurally speaking, it's going to have features that's going to allow it to do its job. The main job of a red blood cell is oxygen transport. It plays a small role in carbon dioxide transport as well, but primary job is O2 transport. And let's take a look at this shape. Okay, a red blood cell is punched in on both sides. All right, it's punched in twice. This shape is said to be biconcave. Bi because twice, and concave, so an indentation is concave, something going, caving inward. And this shape does a lot of things. It gives us a high surface area to volume ratio, which makes it easy for oxygen to get in and get out. It is going to make the red blood cell flexible, which is going to let it twist through skinny little capillaries. It also allows the red blood cells to stack upon one another. And let's look at a picture of such a stack. Here we go. There is a photomicrograph of a capillary. And what we see here are several capillaries in the whole screen, but there is a labeled stack of red blood cells. It's got a little bracket on it, and it's labeled as a rouleau. All right, a little roll, so to speak. And red blood cells stack like this to allow easy passage through skinny capillaries. It's like walking single file through a narrow hallway. It's easy to walk single file. It's hard if everybody's bumping into each other. Okay, so that's the biconcavity. Another cool thing about red blood cells is they're missing something. Look at those red blood cells. Upper right-hand corner, notice there is no nucleus in each cell. They're anucleate. These red blood cells, they're anucleate, and they also don't have organelles. Okay? So the advantage of this is by removing the nucleus, by removing the organelles, there's going to be more room. More room to fill these red blood cells with proteins called hemoglobin that are integral to oxygen transport, as we're going to see in a second. All right, but that's not the only thing. If there's no mitochondria, then there's another advantage. I want you to think about that, and we'll talk about that in class. The analogy I have that's going to help think about it, would you hire this guy, guy who looks like he is shooting up? Would you hire him to move some heroin for you? Think about that and figure out the advantage of red blood cells not having mitochondria. All right. Got another little case to look at here. There's a lady. Her name is Juliet. She is always tired and lightheaded. She has some super heavy menstrual flow. And we got to put these facts together. We'll talk about this in class, but figure out why these things are related. All right, onward, more about red blood cells. We know that they are basically empty, or not empty, but basically bags that are stuffed with a protein called hemoglobin. They're able to be stuffed because they are red blood cells. They are lacking a nucleus and organelles. Now, hemoglobin is a very important protein, and there are going to be billions of hemoglobin. I want to say billions. I have to check the notes. Billions of, or maybe millions. Millions. I'll have to look at it. Or one of you will tell me in class. There are a lot, there we go, of these hemoglobin proteins inside a single red blood cell. Now, what we're looking at in the bottom left there is a single hemoglobin protein. It's made up of these 4 different chains of amino acids. There are 2 that are alpha, the lighter colored ones, and 2 that are beta, the darker colored ones. And so they provide much of the structure of the protein. And within these 4 amino acid chains, there are a total of 4 little chemicals called heme groups. And you can see the 4 red discs in the hemoglobin protein. And a heme group is the chemical, is expanded in the right-hand side there. And we see at the center of the heme group is an iron. So one hemoglobin contains a single atom of iron. And iron is actually what weakly binds to oxygen. Okay? When oxygen has bound to the iron in hemoglobin, we refer to that hemoglobin as oxyhemoglobin. When there are no oxygens bound to a hemoglobin, then we call that deoxyhemoglobin. Now, interestingly enough, carbon dioxide can bind to the alpha and beta chains, to the amino acids of the hemoglobin. And when you've got CO2 bound, that's called carbaminohemoglobin. Remember the rule, the longer the word, the more likely you'll have to write it on a test. All right, so notice that if we have 4 irons per hemoglobin, each hemoglobin can carry 1 molecule of oxygen. All right. Now, oh, by the way, luckily we have about 25 trillion red blood cells in our body. And we've got, a ton of hemoglobins in each red blood cell. So we have a lot of oxygen carrying capacity. All right, let's keep on going. Answer some quick questions here. These are easy. I'll let you do those at your leisure. And let's move on and talk a little bit about making red blood cells. In order to talk about making red blood cells, we got to talk about making blood cells in general. The fancy term for making blood cells is hemopoiesis. Hemo meaning blood, poiesis meaning production, making, synthesis. And it all starts with this stem cell. It's at the top of our flowchart here, called a hemocytoblast. Remember that blast suffix from A and P1 where we had osteoblasts, fibroblasts, myoblasts, etc.? Well, hemocytoblast, literally the blood cell maker, is the stem cell from which all blood cells are going to be made. The hemocytoblast in these arrows here ends up making red blood cells, platelets, all sorts of different types of white blood cells as well. So where do we find these hemocytoblasts? In other words, where does hemopoiesis occur? And by the way, hemopoiesis is sometimes also known as hematopoiesis. And I'm going to show you with some bones. We're going to find hemocytoblasts in red bone marrow. We find red bone marrow in several places in the adult skeleton. We find it in the axial skeleton, so in other words, the skull, the vertebrae, the ribs, the sternum. We also find red bone marrow in the girdles, so the clavicle and the scapula, in other words, the pectoral girdle, and the hip bones, the pelvic girdle. We also find red bone marrow in the proximal ends, the proximal epiphyses, remember that word, of the humerus and the femur. So these are all places with red bone marrow. Now you might wonder, well, why is it called red bone marrow? Well, the thing is, the most abundant blood cell being produced in hemopoiesis is the red blood cell. And so with a lot of red blood cells being made, we end up getting a red color. All right, let's look at a slide of red bone marrow. This is what red bone marrow looks like. These are cells all in the process of becoming blood cells. We do see a lot of newborn red blood cells in there. See if you can pick out a dozen or so newborn red blood cells at least. All right, now the production of red blood cells is known as erythropoiesis. So there's a variety of hemopoiesis. And erythropoiesis happens in the red bone marrow. It starts with a stem cell known as a hemocytoblast. Find that stem cell in the sort of bottom left hand, not bottom left hand, upper right hand. There we go. Several stem cells in this picture. The one we want is in the upper right hand picture. And our stem cell is the hemocytoblast. And that hemocytoblast goes through a series of events, eventually ejecting the nucleus, removing the organelles, synthesizing, hard word to say, synthesizing large amounts of hemoglobin, achieving the biconcave shape, and then entering the bloodstream. That is erythropoiesis in a nutshell. All right. Let's consider a scenario here. What would happen to the atmospheric levels of oxygen as you climb this really high mountain called K2? Well, as you go up and up and up and up, oxygen levels in the atmosphere go down. Because oxygen levels in the atmosphere go down, the oxygen levels in your blood are going to go down as well. You're actually going to deal with this by making more red blood cells. You're going to do erythropoiesis. This is one of the things that causes you to do more erythropoiesis. You're always doing some erythropoiesis. Okay, you have like a level erythropoiesis where you're making, probably, I don't know, a couple million red blood cells every second. Here's a situation where you're prompted to make more. All right, you're going to make more red blood cells in situations where your oxygen levels decline. Now, the organ that senses the decline in blood oxygen is the kidney. There's the kidney right there. In response to a drop in oxygen levels, the kidney releases more of a hormone called erythropoietin, which is sometimes abbreviated EPO. Erythropoietin travels in the blood to the red bone marrow and stimulates erythropoiesis, which in this case is going to cause you to make more red blood cells. All right? And by making more red blood cells, you're better at carrying oxygen, which can kind of offset the initial problem, which is not having as great of levels in the blood. Okay, so let's backtrack for a second here. The production of red blood cells is called erythropoiesis, literally red making. It occurs in your red bone marrow, which we got to know the bones where we find that. Erythropoiesis is something you're always doing. You're always doing it. You're always causing hemocytoblasts to undergo changes, to differentiate, to eventually lose their nucleus and organelles, to make lots of hemoglobin, become biconcave, and turn into a red blood cell. You're always doing this. There is a hormone that stimulates this process. That hormone is called erythropoietin. You're always making some erythropoietin. One situation where you're making more erythropoietin is the one we just discussed, which is when blood oxygen levels decline because of a change in atmospheric oxygen levels. There could be other causes of blood oxygen levels declining, certainly, and all of them would prompt an increase in the production of erythropoietin. And erythropoietin is made by the kidneys. It's a hormone, so it travels in the blood to the red bone marrow, causes red bone marrow to do more erythropoiesis, and we make more red blood cells. All right, I got a couple pages here, a couple screens, slides, I don't know, whatever you want to call them. Suppose you started smoking. Suppose you started smoking cigarettes. You took this doctor's advice. You got to love this ad. I don't know when this is from, maybe like the 50s or something. I don't know. So when you smoke cigarettes, that's actually something that's going to cause your oxygen levels in your blood to decline. So blood oxygen declines. I want you to figure out what would happen to these 6 things and explain why. And I'm going to ask you guys about this in class. And then, once you've done that, that's like the warm-up. Then I want you to do these 5 questions here. And I'm going to ask you all about these in class. Then when you're done those, I want you to do these. Maybe we'll do these in lecture, maybe recitation. I don't know. Figure it out. All right, so what we've discussed a little bit so far is the production of red blood cells, erythropoiesis, the structure of red blood cells, the biconcave shape, the lack of nuclei and organelles, the basics about hemoglobin, the hemes, the iron, etc. What we want to do now is move into the destruction of red blood cells. And a good thing to talk about in terms of red blood cell destruction is a bruise. So you get hit, all right? You get hit in the leg, the arm, or something, and you get a bruise. What's going on there is blood vessels are breaking underneath the skin. Blood is spilling over into the normal dermal space. And you're going to see color changes. Maybe the bruise starts out as reddish, turns purple. Eventually it may go through a whole like shades of yellow and greenishness. It's really good bruise. So what we're seeing is what's going on with red blood cells, okay? When those red blood cells spill into the tissue space, we got to deal with them. They need to be destroyed, and the parts need to be recycled or reused. So what's going to happen is macrophages are going to come to the site of injury. Macrophages are going to engulf red blood cells and start the process of destroying them. Now, excuse me, in the picture on the bottom left there, we see a macrophage that has swallowed 3 red blood cells. See if you can find out. Now, we know red blood cells are basically just bags of hemoglobin. So what we're really figuring out in red blood cell destruction and recycling is what happens to the hemoglobin. Remember, there's 2 parts to the hemoglobin. There's the heme, which is the pigment that had the iron. We're going to recycle and get rid of some of that. Then we got the globin, the amino acid part. That's totally going to be recycled. From the macrophage, those amino acids are going to end up back in the blood for reuse somewhere else. For some, make other proteins, broken down for energy, whatever. However, the heme is a different story. What we're going to do to that heme is take out the iron. We got to recycle the iron. Iron is a valuable commodity. And the iron, excuse me again, is going to be carried by a plasma protein, a globulin, called transferrin. Literally means iron across. And the iron is going to be carried to the liver. And we store the iron in the liver. And we store it as some protein complexes. There's one called ferritin, one called hemosiderin. Iron is kind of toxic. It's just floating around by itself, so you don't want to do that. Then ultimately, the iron needs to go back and be incorporated into your muscle cells or back into red bone marrow to make more red blood cells. Okay, so that's the recycling of iron. The rest of the heme needs to be gotten rid of. And when we take the iron off, we turn the rest of the heme into a compound called bilirubin. And bilirubin has a really yellowish color. It's what accounts for the yellowness of a bruise. And bilirubin is going to be carried to the liver as well by good old albumin. And then in the liver, bilirubin is going to be modified. It's going to be secreted into the small intestine as part of a digestive fluid you may have heard of called bile. And then the bilirubin gets metabolized by bacteria in the colon, the large intestine. Some of the metabolites go back into the urine and get peed away. Sorry, go back into the blood and then into the urine and get peed away. Most of the metabolites from bilirubin end up getting pooped away. And that is what accounts for the normal color of poop, is the breakdown products of bilirubin, thus of heme, thus of red blood cells. So a little insight into red blood cell destruction can be found in looking at the life cycle of a bruise. Now, excuse me again, most of the time, you are not destroying red blood cells just as a consequence of bruising. Most of your red blood cell destruction occurs in your spleen. Millions of red blood cells are destroyed in your spleen every second. Your spleen is a red blood cell graveyard. Its main job is to look for old and ugly red blood cells. The spleen has some very twisty capillaries, which are hard for old and damaged red blood cells. Old ones and damaged ones may not be flexible enough to get through. They're going to be noticed when they're stuck. Macrophages are going to come up and say, hello, you're old, you're ugly, I'm going to kill you. And they're going to eat them up and do the basic process that we just went through. All right. By the way, if you lose your spleen, that red blood cell destruction job falls to your liver and to a small degree to your red bone marrow as well. Let's talk about bilirubin a little more. Bilirubin is a normal breakdown product of red blood cells. It is a yellowish pigment. Sometimes people can have problems getting rid of bilirubin, or making too much bilirubin for that matter. And the skin, the sclera, the white of the eye, can take on a yellowish color. People can become jaundiced. Now remember, bilirubin is secreted as part of a digestive liquid called bile. So if you have gallstones that are blocking a bile duct, like what's pictured on the left there, it is going to be harder to get rid of bile and thus get rid of bilirubin, and it can build up in your tissues. If you're an alcoholic, you're damaging your liver. Well, your liver can't metabolize the bilirubin the way it normally would. And you can become jaundiced that way as well, because the liver can't deal with the bilirubin that is being brought to it from the spleen by albumin. Now, maybe most familiarly, babies can often have a physiological jaundice when they're born. The thing about babies is babies are breaking down a lot of hemoglobin because they're actually switching from a fetal to adult hemoglobin right around the time of birth. And so you got all this breakdown of hemoglobin that's happening, but the baby liver is so little, can't handle it. So bilirubin can build up, which can be problematic if it's not dealt with. But of course, there are means to do that. All right, thus ends our discussion of bilirubin and red blood cell destruction. All right, we're going to talk about white blood cells now, and that's going to finish up today's, or not today's, this particular lecture. White blood cells. There are maybe 5 to 10,000, 4,500 to 11,000 per microliter of blood. And I want you to contrast that with the fact that there are about 5 million red blood cells in a microliter of blood. Huge difference. Huge difference. So not a lot of white blood cells, relatively speaking, in the blood. They're primarily using the blood as a transport system to move to get from place to place in the body. Now, you've already done probably, or I guess it depends on when you're listening to this. Maybe you have, maybe you haven't done this yet. But there are 5 basic types of white blood cells. There are neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Never let monkeys eat bananas is a good mnemonic to help you remember those names. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. Once you've had the lab on blood cells, you should be able to identify all 5 of the white blood cells in that picture on the left. Now, your white blood cells, primarily the most abundant white blood cell in the blood, around 60% are neutrophils. Around 30% of your white blood cells are lymphocytes. Around 6% are monocytes. Around 3 are eosinophils, and 1, maybe, if that, are basophils. Okay, let's talk a little bit about white blood cells in general, and then about these different types of white blood cells. So if white blood cells are not that abundant in the blood, well, where are they? We have trillions of these in the body. They're just not in the blood. Major places are in places where they encounter pathogens, bacteria, viruses, microbes, whatever. So where are we going to find them? We're going to find them in our tonsils. That's the upper left-hand picture. We're going to find them in the lining of our trachea. That's the upper right-hand picture. In our lymph nodes. So we see some lymph nodes in the axillary region on the lower left picture. And also in the lining of the intestines. That's one of the most abundant places. That's the bottom right-hand picture there. So we're going to see them in places where we're going to meet pathogens. All right. White blood cells are made in a process called leukopoiesis, which is just another type of hemopoiesis. So white blood cells are also known as leukocytes, white cells. Leukopoiesis, white making, happens in your red bone marrow in the usual places. It's part of hemopoiesis. It starts with hemocytoblasts, the stem cell. Remember, all blood cells start with hemocytoblasts. And a lot of things go on. A lot of stuff happens. Eventually, from that hemocytoblast, we make the different varieties of white blood cells. Now, white blood cells, as a group, are able to battle pathogens, to fight infection. And there's a couple characteristic things they can do that make them really good at this. 3 that we got to hit are diapedesis, migration, and positive chemotaxis. Diapedesis is the fancy word for the process by which white blood cells get out of blood vessels. White blood cells have the ability to converge and amass themselves at sites of injury. They get there via the bloodstream. However, they got to get out of the bloodstream. They got to get through the epithelial lining of the blood vessels of the capillaries. And that squeezing through in between the capillary simple squamous epithelial cells that we see here, upper right-hand picture, that is called diapedesis, which is a fun word to say. Okay, then once white blood cells leave the blood vessel itself, they got to be able to move. They got to be able to sort of wriggle along or migrate. And they can do this to get to the damaged area. But you got to ask yourself, well, how do they know where to go? And what they do is they follow a gradient of chemical cues. So damaged cells, pathogenic cells, release chemicals that attract white blood cells. White blood cells will move towards where the quantity, the concentration of those chemicals is the greatest. And that movement towards a particular chemical is called positive chemotaxis. Taxis means movement. So we have movement towards chemo, a chemical, because it's towards, it's positive. Negative would be away. All right, let's go through our different types of white blood cells. We did a lot of this in lab, so it is kind of a reiteration here. Two big classes of leukocytes of white blood cells, the granulocytes and the agranulocytes. They're based on how they look under a normal microscope. Under a normal microscope, granulocytes have little granules that are visible. There are three types of granulocytes: neutrophils, basophils, and eosinophils. Agranulocytes, on the other hand, lack the visible granules under the light microscope. When you treat white blood cells with this dye called Wright stain, you can see granules in one group, but you can't see them in the other. Two types of agranulocytes: monocytes and lymphocytes. All right, let's review the five guys here. We're going to start with the neutrophil. There's a neutrophil on the right-hand side there, upper right. Neutrophils generally make up around 60%, maybe more like between 50 and 80, but around 60% of your white blood cells in your blood. They are phagocytic. They are excellent at destroying bacteria. They are very abundant in acute bacterial infections. Like if you had appendicitis, there's a removed, ruptured appendix with some dead appendix tissue in it up there. So if you had appendicitis, if some poop blocked the opening to the appendix and bacteria built up, and you got appendicitis, and the appendix got inflamed, neutrophil numbers would rise like crazy to deal with it. So neutrophils are very phagocytic. They are bacteria killers, roughly 60% of your circulating white blood cells. They have a multi-lobe nucleus that we see very clearly there as well, which makes them really good at diapedesis. They have granules, but they're not super colorful, kind of like a lilacish color. All right, that's one granulocyte, two more to go. There's a beautiful eosinophil right there. You can see it, the one that my crosshairs are on. And the eosinophil often has a bilobe nucleus, and it is going to have these distinctive reddish-orangish granules. And these granules are filled with chemicals that are anti-helminthic. They're very good against parasitic worms and flukes. We see some parasitic little wormies embedded their mouth faces into the wall of the intestine. We see a long worm, was that a guinea worm, I guess, being removed from somebody's foot very carefully and slowly. So eosinophils are the cell that we use to try to kill parasitic worms, parasitic flukes. They're also involved in decreasing the severity of immune responses against things that are not harmful. In other words, they try to decrease the severity of allergic reactions. Obviously, eosinophils are not always successful in either of these two jobs. Usually about between 1 and 3% of your circulating white blood cells are eosinophils. Now check out the ad on the bottom right here. Pretty funny. All right, let's keep going. Last but not least, of the granulocytes, the basophils. There's a nice basophil right there. Notice the nucleus. We can see the nucleus. I'm kind of tracing over it there. The nucleus is visible, but so are lots of little purple-ish dark granules. These granules are filled with chemicals involved in inflammation. Chemicals like histamine and heparin. So if you're touching poison ivy, so you see some poison ivy, you don't know that, leaves of 3, let me be, you don't know that little rhyme, you pick up the poison ivy and you rub it in your hands. You're going to have an inflammatory reaction. Basophils are going to release histamine and heparin, which are going to cause vasodilation and a whole lot of itchiness. Now, inflammation is actually a good thing. I mean, not so good in the case of poison ivy when it goes overboard, but in almost all cases, it is a very good thing, okay? And basophils help mediate the process of inflammation. Usually somewhere between 0 and 1% of your circulating white blood cells are basophils. Doesn't mean you don't have them. They're just not in the blood that often. All right, 2 types of agranulocytes. Let's do those. First and foremost here, we've got the lymphocytes in that square right there, or rectangle, I guess. I'll let you guys measure it for me. Let me know. There is a lymphocyte. Lymphocyte usually has a kind of a medium purple nucleus, no visible granules. Lymphocytes are of 2 varieties. There are T lymphocytes and B lymphocytes. Getting some background music here. I wonder if you guys can hear that. We'll see. T lymphocytes and B lymphocytes. T lymphocytes are very good at killing cells that have become infected with viruses. And we see a virus on the upper left up there. T lymphocytes also control and coordinate your immune response. B lymphocytes are the ones that end up making antibodies, special immune proteins. There's actually another specialized type of lymphocyte that we'll talk about when we deal with the immune system called the NK cell, or the natural killer cell. All right. Let's keep on going. If you have an acute viral infection, your lymphocyte count is going to go up as you try and deal with it. And we see in this slide of blood here a lot of lymphocytes. See if you can pick those out. Now we also have one more cell to do. The last cell to do is the monocyte. Look at it up top there. Look at its U-shaped, so it's a kidney shape nucleus. Look at its pale cytoplasm. Monocytes are in the blood, and when they leave the blood, they change and they become macrophages. Now, monocytes are not involved in mononucleosis. Don't believe that rumor. Monocytes typically make up, about 6%, maybe 5 to 8% of our white blood cells. Back to lymphocytes for a second. Lymphocytes normally around 30%, maybe between 20 and 40, somewhere around there. All right, one more thing to talk about. What does this look like here? Does this look like leukocytosis or leukopenia? Look up those 2 words if you need to. Come back and tell me, and I will see you guys next time.